So I'm going to talk today about natural immunity, and I'm going to tell you what that is. And specifically, honestly, I'm going to focus on one cell type, the natural killer cell, that is fairly neglected. And I hope that I can convince you by the end of this that they shouldn't really be as neglected and that they're potentially really interesting. But I wanted to begin with a little bit of background because I know it's been a while since, I thought this was working. Okay, since folks have done this, and I don't know how many, we've probably got a diverse audience here, and I tend to throw around terms, and immunologists are famous for using a lot of jargon, which I think is probably the worst trait of my profession in terms of my research lab, because it makes it less approachable to other people. But there's no reason for all the jargon. Nonetheless, I will slip into a few. So first of all, let's just define immunity. And immunity is defined, according to Google, really, as the ability of an organism to resist a particular infection or toxin by the action of specific antibodies or sensitized white blood cells. So immunity, I mean, there's also immunity in a legal definition, of course, which comes up these days. But I'm talking specifically about a body and something that we don't want there, and it's our ability to resist that. An antigen is one of these jargon immunology terms, but all it means is a toxin, so something bad for you, or another foreign substance, which induces an immune response, so it induces that immunity. And of course, you can see already this very antibody-focused view of the immune system, which I'm going to try and break down, especially the production of antibodies. And that's important because antibodies protect, and I'll come back to that. Now, you can get more specific. So an antigen can be anything foreign. It can also be, that pollen that you're allergic to, which is driving you to sneeze today. But it can also be an infectious agent. A pathogen, on the other hand, is a disease. It's a bacteria or a virus or a fungus, any other microorganism that causes disease. And so that's important because right now we talk a lot and hear a lot about our microbiome. We're more bacteria by cell than we are human cells. But most of those are what we refer to as commensals. They're good bacteria. They live with us. They're fine. But a pathogen is one we don't want there. And of course, I think people do get confused about bacteria versus viruses. So just to be clear, a bacteria is an organism. It's a large group of unicellular organisms, so one little tiny cell. And they have cell walls, but they don't have any fancy internal structures. And some can cause disease, and some can be those commensals that hang out with us. And a virus is arguably not alive. It is an infective agent, which is defined as it can spread itself. And it consists of nucleic acids, so RNA or DNA, usually in a protein coat, sometimes with or without an envelope. And it's too small to even be seen by conventional light microscopy. And it's only able to multiply in the presence of a host cell. And so that's the key, is the virus on its own is totally useless because it's just a particle there. But if it gets into the cell, then it takes over some of the host cell machinery to replicate itself. And that is an important distinction for what I'm going to get into later because the type of cell I'm interested in tends to worry about things that are inside cells, not things that are outside cells. Most bacteria are outside cells. Most viruses are inside cells. They're, of course, well, all viruses are inside cells. They can pass between them, but they don't replicate outside cells. So everyone clear? Yeah? We got some definitions? So now it's going to get easier from here, or not. We'll see. So what is natural immunity? So this is perhaps the most re-borrowed slide in immunology right now. If you just Google immune system, you'll get this slide. It's from a really nice review in Nature Reviews Cancer back in 2004. And it really shows very nicely the idea that immunologists break the immune system into two major components. We've got our innate immune system here, over in this big circle, which is what I refer to as our natural immune system because it is what naturally protects us from things. And then on the other side, we have our adaptive immune system. Now, I'm going to go over this a little more. So the innate immune system is generally thought to be kind of non-specific. So these are parts of your body that in a not pathogen specific, so it's not like flu versus HIV, it's like general features of virus, I'm going to respond. And that's in contrast to the adaptive immune system, which is highly specific. And the way B cells and T cells over here work, and again, I'll go over this in a little more detail, is they actually recognize tiny little pieces of an individual pathogen in a highly specific fashion. And so just to talk about a few of these cells to put it in context. So up here we have our macrophages and our dendritic cells, and these are your phagocytic cells. Phago is a Latin root for, anyone know? Very good, yeah. So what do we suppose they do? They probably eat things, right? So literally they cruise around and they eat up any bacteria or viruses or even disease cells that they run across. And then actually, they not only eat them, they throw stuff from those stuff on their surface and they talk to the T and the B cells and they say, do something about this stuff I just ate. So there's a lot of crosstalk here. But their main function is eating stuff. Now we've got some other cells here, the mast cells, basophils, eosinophils. These can often be annoying cells for those of us in modern society because they mediate a lot of allergy. But they probably exist not to make us miserable with sneezing, instead to fight worms, which are a thing that happens. We get intestinal worms, and they secrete all these annoying cytokines, which are chemical mediators, that make your gut contract and expel worms, which is great if you have worms, less fun if that's happening in your lung. So there you have it. So these are those guys. They have many more functions than this, of course. I'm massively oversimplifying it, but there you have it. Neutrophils are the first vanguard of your immune system. They're the first line of defense. They only live 7 hours on average. So you're constantly making these buggers all the time, but if you've ever had a pussy cut, that pus is actually just a bunch of dead neutrophils because they're the guys that go in, they're also phagocytic, they eat lots of stuff, they kill lots of stuff, they cause mass havoc, but in a generally good way until it gets out of control and you end up with big nasty pus pockets. There's also various proteins, complements on this, but there are many sort of naturally anti-pathogen proteins that act in a variety of different ways. And then finally, over here we have natural killer cells, which I'm going to talk about a lot because I think they're really neat, but which have the natural ability to kill other cells that they perceive are a threat. So they recognize altered self, and I'll go into the mechanisms of that in much more detail with a little bit of framework. But I want to get back to now the adaptive side. And the reason the adaptive side is so important is because it is the side that is thought to have immunologic memory. And memory in the immune system isn't that hard to figure out. It's defined as a functional response, the ability to respond better and faster the second time you see something. And what happens is when you've been exposed to something before, your B cells and T cells are slow to respond. They don't even get into the party until 3 to 7 days later, after all your innate cells have been involved. But when you get re-stimulated, they're going to respond instead in 2 days or 1 day. And there are already more of them, so you're going to respond better. And then we've got our antibodies here. I realized I forgot. And the antibodies are preformed. And in fact, your antibodies hang around in your bloodstream for a while and can protect you by binding up the virus or the bacteria or whatever and signaling it to your immune system, either directly inactivating it itself by preventing it from entering a cell or by tagging it for those phagocytic cells. Because it turns out the phagocytic cells will eat up antibodies that are bound to something. So it's kind of a neat system. And then we've got our T cells, which coordinate the immune response and really tell the B cells what to do. So the B cells make the antibodies, which are awesome, but without the T cells, you don't make enough B cells and you don't have any antibodies. So you need them both. And then there are these kind of weird cells that are in the middle, which are sort of T cells, but sort of innate because they don't have very specific receptors. They have receptors for particular features of really important pathogens in human history, like tuberculosis or things like that. And so they're narrow in their scope. So now I'm going to talk actually randomly about vaccines because, of course, the reason we get vaccinated is in order to protect us better the second time we get something. That's why hopefully we're all contemplating getting our flu shot right about now because this would be flu shot season. And just as a review, and this will all make sense later, hopefully, I'll bring it back around. Vaccines work great. And the way it works is you basically take one of these pathogens and you either take a chunk of it so that it can't replicate itself or you weaken it in some way and you give it to somebody. And the weakened forms hopefully won't cause disease, although if anyone read the New York Times this morning, actually there was an interesting article about polio and how some of the polio live vaccines have mutated to cause disease, which is not ideal. But preventable, ironically, if you use the other vaccine. So you give a weaker dead portion, your T cells get activated, they talk to your B cells make antibodies. And the antibodies are there to inactivate that bacteria or virus or whatever it is. And so if you get reinfected, you've got those preformed antibodies. Now, the antibodies hang around for quite a while, months. And in fact, 3 to 6 months per individual antibody, but they're different cells making them all the time. And that's exactly why we should be getting vaccinated right now for flu. Because you begin to make antibodies at about 7 days post-vaccine. You reach your peak response at about 28 days post-vaccine. It maintains at a pretty high level for another month or two, and then slowly tapers off. And then you've got all those antibodies hanging out there. And they will protect you through the flu season, which typically will tail off about March. And so that's why we time the flu vaccine. Makes sense, right? Now, this is a great system, and I would argue that vaccines are probably one of the single medical interventions that has saved the most lives. Now, I'm not talking about public health interventions. Clean water has saved the most lives, but from medical intervention. But of course, there's a lot of controversy right now. Happy to take questions at the end. But what I want to focus on is, what about diseases for which we don't have a vaccine? Or diseases where, annoyingly, we have to get the shot every year, right? Nobody likes to get their flu shot every year. Wouldn't it be better if we had one flu shot that worked for 5 years? Would be nice, right? And so to sort of put this in perspective, I'm going to talk about HIV, which is the quintessential example of where we've been working for 30 years, unfortunately so far unsuccessfully, to make a vaccine. And the first thing to think about is why we even need a vaccine. And this slide is a little outdated, but this is from 2011. Estimates made in 2012 by the UNAIDS and the World Health Organization, and it shows the burden of HIV across the world. And you can see that at that time, there were about 1.4 million people in North America, but 23.5 million people in sub-Saharan Africa living with AIDS. And you can see it's not evenly distributed, but there's a lot of people living with a problematic disease. What's more concerning is, so unfortunately UNAIDS doesn't make the pretty maps anymore. So I use the old one. This is the most recent statistics and estimates. So we've gone from 34 to 38 million people living with HIV. Now, this is a multifactorial thing. We have more people living longer because we've got great drugs, and that is fabulous. But if you look at the bottom, we've got 1.7 million people annually becoming newly infected. And that, I think, is a problem, and that is a problem that arguably we could or should prevent. And there are a lot of children still being infected despite the fact that we know how to arguably prevent mother-to-child transmission. And so would a vaccine really make an impact? And this actually is an arguable point, but again, this is just one example of a vaccine. But we've got absolutely astoundingly good antiviral medications. And they're very effective. Availability is more of an issue. But they're effective not just in treating people who are infected, but in preventing new transmissions. And they do this 2 different ways. The first is that someone who is infected, if they're on effective antiretroviral drugs, essentially does not transmit the virus. If their virus level is suppressed in their bloodstream with the drugs, which if they're on appropriate therapy, it should be, there is a 99.9% chance they won't transmit to their partner. So that's pretty effective, right? If we just got everyone who had HIV on drugs, we would end this epidemic in a generation. Absolutely. In addition to that, people who don't even have it, but who are at risk, if they take the drugs as pre-exposure prophylaxis, they also don't get infected. So it's shockingly effective. So if we just manage to put antiretrovirals in the worldwide water supply, it would be over. But of course, do we really want to do that? So the issue is the drugs are incredibly costly. They do have side effects, some of which can be serious. We also see emerging drug resistance because we use a lot of the drugs, and so they're effective against, but not against a drug-resistant strain. And the most underappreciated, but arguably most important, is the identification of risk groups. So there have been several trials of these pre-exposure prophylaxis, and the first couple were amazingly effective. And they were done primarily in the US among men who have sex with men, and everyone took their drugs and they were 99% effective. And then a study was done in sub-Saharan Africa, and it showed no efficacy. And a lot of names were called and unpleasant things were happening that clearly they didn't know how to run a study, whatever. All the participants said they were taking the drugs. But when they went back and did blood testing, pretty much no one took the drugs. And there's a lot of, and then years of study went into this. So there's huge social stigma in being caught with antiretroviral medications in sub-Saharan Africa because people think you're infected. So nobody who's healthy wants to get caught with drugs. They also have side effects. And if you feel fine and you don't perceive you're at risk, you're not gonna take the medicines. And if you don't take them every single day, they don't work. The estimates from the US studies were that you had to be 88% compliant for it to be effective. That's pretty good. Who can remember to take a pill every single day? I mean, even birth control, I don't think people are that good, and that's another thing. So I'm not gonna say much about this. Education-based prevention. People should just be cautioned to use condoms. That's worked really well for the last 40 years. So what about vaccines? So we've had 3 really large scale trials. And there have been glimmers of hope, and actually this year may well be a banner year. There's some really exciting studies coming out. But up to this point, it's been pretty grim. There was a study in 2003 called the VaxChin trial, absolutely no protection. There was another study referred to as colloquially as the STEP trial. It was a Merck vaccine, 2007, no protection. And that's being kind because actually in certain subsets, it increased the risk of HIV acquisition. So unsurprisingly, that set the field back for a few years. Luckily, the RV144 Thai trial was going on in parallel. So it didn't get stopped and was a totally different method. And it did show some glimmers of protection, but it was very low, 31%, and it was very short-lived, meaning the protection only lasted 6 months, and this is a disease we're constantly exposed to. But it provided enough of a signal that a bunch of follow-up studies have been ongoing, and these things take time. And so I do think in the next couple of years, you'll see something. But just to put that 31% in perspective, anyone care to guess what the efficacy of a single dose of measles vaccine is? 90, we got it, 90. And 2 doses is more than 99, or 97, depending on which study you read. But either way, it's really high. Okay, so 31%, not so good. So a big question is, why is this so hard? And to explain this, I want to talk a little bit about HIV variation. And this is the same issue or a similar issue for flu and for dengue and for a lot of other diseases that we study for which we don't have a vaccine. And to do this, I'm going to show a phylogenetic tree. Has anyone ever seen or heard of a phylogenetic tree? We got a couple people. Okay, I'm going to explain it as a family tree because it's really just kind of the same thing. So here's a family tree. We've got a grandfather, grandmother, aunts, uncles, mothers, fathers, brothers, sisters. And what you can do is, with a tree like this, you can figure out how related people are by the distance between them. There's like branch links. So this person is further from this person than this person is, right? Not that hard. So you can do the same thing with viruses. And you can just say different virus strains and look at their branch length difference and see kind of how diverse the population is. So it's not a perfect analogy, but it's what I got. So a few years ago, Betty Korber put together this, who's a brilliant informatician at Los Alamos, put together this nice paper to sort of put in perspective the HIV issue. So in this, and I'm sorry that it looks so terrible, but this is unfortunately as good as it gets in the original paper. From 2001, so before the PDF era. So she put together a phylogenetic tree of 96 influenza sequences from 1 year. And again, we kind of all get the sense that flu varies a fair bit from year to year and even within year, because that's why we have to get that darn shot every year. So you can see there's a scale bar, 10% difference. So there's a fair bit of variation in that 1 year. This is 9 strains of HIV from a single person who's been infected for 6 years. right? So what you're noticing, I hope, is that there's just as much variation in one infected person as there is in 96 different flu-infected people. And that's because the virus introduces basically a mutation every replication cycle. And if you think about 1 billion particles introducing one mutation every cycle, you can end up with a lot of variation. Even if a bunch of them can't make it, enough of them do that you get a lot of variation. And hopefully you're guessing what's going to come next. This is HIV in one country, in the Democratic Republic of the Congo, which admittedly is where there's the most diversity of HIV because it's nearer the places where it arose. But what you can see is these are, again, all strains from a single year, 1997, 193 strains. And you can see this tremendous diversity. So if you look at the branch links, this might as well be a whole new virus compared to this. And in fact, they're broken into groups that we call subtypes or clades. And they're about 30% different from each other between clades or subtypes. So when we're talking about, gosh, we should be able to come up with a vaccine, do we need one vaccine or 193 vaccines? Right? It's not that easy. And so the problem with the VaxGen and all the other trials was you got type-specific responses. You know, the one strain that we used to generate the vaccine, we could protect against that, but it doesn't matter when you're facing this massive gemisch of all these different strains. So one of the things, I spent many years doing the neutralizing antibodies and T-cell work. And a few years ago, we decided maybe it was time to think outside the box about how to tackle this and focus on some other cell types to get around this diversity problem. And this is how I became interested in NK cells. So I promised you would come back, and slowly I've come back. And so the question is, why natural killer NK cells? So if we go back to our schematic, just as a reminder, they're here. They're classified as innate immune cells because they naturally kill things. So this is just a picture of some NK cells. Well, an NK cell and a monocyte, one of those other cells that was on that picture. My postdoc took these pictures. He dyed the NK cells blue and put something marking the rim. And you can see that it's next to another cell, a different kind of cell, the monocyte from the same individual. And you can see the NK cell is kind of a boring round cell. You know, it's just nothing very exciting about it. And it doesn't have the beautiful antigen-specific receptors that a T cell or a B cell has to say, that's a flu-infected cell, that's a HIV-infected cell, whatever. But this is a picture of NK cells from that same person and monocytes from that same person. But what Mathieu, my postdoc, did before he mixed them, oh, you can't even see the monocyte. Trust me, there's another cell there that is he infected the monocyte with flu. And without any specific receptors for flu, somehow this NK cell knew that it should react to that monocyte. And it's no longer round and boring. It's kind of ruffled, and it's polarized, and it's long and skinny. And it's formed this contact point between the two cells, which is referred to as an immune synapse. Just like a synapse in our brain, it's a communication point between the cells. Only the NK cell synapses are a little more nefarious than the immune synapses. They use the synapse to punch holes in the cell next door and introduce toxins into it and kill that cell. And in fact, oh, I'll show it later. I was thinking it was the next slide. So really one of the big questions is, how does this work? And could we figure out how to control it? Because in theory, it should work for any flu strain or any HIV strain if we knew the rules of engagement. So the fun thing about NK cells is that they actually are kind of new to the scene as immune cells. They weren't actually even reported to exist until 1975. There were back-to-back papers in the mouse system and in humans reporting this weird natural cytotoxicity, meaning cytocell toxicity killing. So they could kill tumor cells, and nobody could understand why, because they weren't T cells, and everyone knows about T cells. And it wasn't until 9 years later that Louis Lanier, my colleague up at UCSF, and some of his colleagues wrote this opinion piece saying that this activity that was reported actually refers to a cell. And I propose that we call this cell a natural killer cell. So thanks to Louis, we have natural killer cells, which is kind of a fun name. But the question is, do they even matter? Because they're kind of rare. It took a long time for people to discover them and whatnot. But just a few years after Lewis wrote that perspective, Christine Byron published in the New England Journal of Medicine this paper reporting severe herpes virus infections in an adolescent without natural killer cells. So by this time, they'd figured out that there was a specific phenotype, a specific set of markers they could use to identify NK cells. And there was this kid, and here's the story. A 13-year-old girl presented in 1984 to UMass with life-threatening chickenpox infection. And she was treated, and then they reviewed her history, and she'd had all these weird recurrent infections, mostly with herpes viruses. And here I'm referring to herpes virus as a class that refers to herpes itself, cytomegalovirus, Epstein-Barr virus, which causes mono, and chickenpox. And so they thought this was funny, and they'd been like looking and trying and studying her T cells and whatever and trying to figure out what was wrong, and Christine was smart enough to notice there were no NK cells. And 5 years later, they'd figured out that maybe there was something wrong, and so they did this. This is the penultimate proof that it's NK cell activity. So these are killing assays. What we're looking at is the ability of people to kill these tumor cell lines, and this is a killing that is not T cell dependent, it's NK cell dependent. And what you can see here is that all the bars that are positive are different tumor cells, and they grabbed like, however many this is, 20 healthy people, and they could all, to some degree or another, kill the tumor cells. These cells are a little more resistant, these cells about the same. Here's our patient, really can't kill, and even when you add interferon to boost this response, it's just a way to boost a response, they still just don't really respond. So clearly they matter for herpes virus. Whether they matter for anything else, we're still figuring out. But in fact, since then, and now going back, the mutation that caused this has been identified, and it's actually not on this list, but there are a whole bunch of different mutations that are associated with NK cell deficiencies. And if we just look at what happens, it becomes clear that you need NK cells for herpes viruses, because all these mutations that screw up your NK cells cause herpes viruses, herpes viruses, herpes viruses, herpes viruses, or herpes virus infections. So there are some other things that happen, and in fact, this is not nearly complete. There are many, many more with a partial defect. But it's also important to know that there's a lot of overlap, and there are defects in other cell types in each of these. So it's not entirely clean. But it really says that NK cells are critical for at least this part of our immune system. Okay, so to sort of go through this, so how do they do this? And it turns out they're really critical in that early innate response as the first line of defense against viral infection and other intracellular pathogens. So the first thing that happens is your phagocytic cells secrete some cytokines that activate lots of things. And then very, very quickly, within a day, you're starting to get your NK cells coming up. And this is our virus level. And then it's not till day 3 or 4 and up to 7 that we start to get these are the T cells that kill virus-infected cells. So the NK cells are important in that first stage, and then they also talk to these guys. So how do they do this without any ability to say, I know what flu is, or I know what HIV is? And the way they do it is that they have a balance of what we refer to as activating and inhibitory signals. So this is not as hard as it could be. So normally in healthy cells, we've got our little NK cell here, and it's got what we refer to as activating receptors on the surface. And it turns out what these activating receptors do is they recognize stress on the part of another cell. So when another cell is stressed, it will send signals. But they're also, and this is critical, inhibitory receptors. And all these inhibitory receptors recognize a molecule called major histocompatibility complex type 1, or MHC type 1. In humans, annoyingly, back to jargon, it's referred to as HLA, or the human leukocyte antigens. You may hear any of those terms. They all mean the same thing. The important thing to know is that it's on every normal healthy cell in your body except red blood cells. So basically what happens is if you have another cell, that cell should express MHC class 1 because that's what cells do, and maybe some low level of stress-related signals. But normally, and those are activating ligands and MHC, normally what happens is the inhibition wins over the activation, and the cell basically tells the NK cell to bugger off. I'm a normal healthy cell, leave me alone, and the NK cell goes about its way and doesn't kill that cell. And you need this because I just showed you they could kill things. We don't want them going around hairy-carry killing everything. So that means there must be activation scenarios. And these aren't very hard to figure out as long as you think about this tipping the balance. One is that a lot of viruses and a lot of tumors will down-regulate class 1 expression because class 1 turns out to be the way that T cells recognize them. And T cells are really bad if you're a virus or a tumor because they can kill you. So they try to escape from that, but that can lead to an unopposed activating signal. And so this is a backstop. So the NK cell can kill that. Alternatively, you can still retain class 1, but the cell can be so stressed that there's enough activating receptors to win, tip the scales towards activation, and lead to NK cell activation. So because everyone likes a movie, let's watch it in action. Green cell is a tumor cell, or sorry, green cell is the NK cell, red cell is the tumor cell. And what we're gonna watch is the NK cell pushing and prodding on that tumor cell, making this beautiful immune synapse, it's actually probably already made here, and then injecting stuff into that cell and killing it. And you're gonna see something happen, which is that there's what we refer to as a viability dye. It's a blue dye, and when that blue dye appears, that tumor cell is dead. And you can see the cell is kind of swelling before that happens because it was injecting stuff into it, and now the tumor cell's dead. And so this is a really dynamic process, right? The NK cell is sort of pushing and prodding, and that's probably all those different receptors being received and getting ready and killing that cell. And you can see the time scale up there. This is happening in about 40 minutes. And in fact, that's slow. They did this, my colleagues did this at room temperature because they wanted to slow it down to watch what happened. At 37 degrees of body temperature, the NK cell can kill, disengage, and go to the next cell in 5 to 10 minutes. And in fact, this also brings up a fun thing, which is the phenomenon of serial killers. It turns out that there are relatively in any given culture, and we don't understand why, there are a few NK cells that are particularly good at killing. And they'll go around, like some of them are just kind of hanging around lamely, and the other ones are just kill, 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 right? So that's fun. So for more stupid movies, my grad student made this one to explain that I've talked a lot about killing, and NK cells do kill, but they do more than that. They can actually do a lot of important things. But this is just a claymation demonstration for all those of us. So what I've been showing you is direct cytolysis. It's literally punches holes in the cell and puts things in it that digest the cell from the inside out. And so we can see that our NK cell has killed the target cell and is very happy with itself. But there are also receptor-ligand interactions that can induce death. And I'm sorry, I don't know why that one didn't appear. And so we can see that that's another way to kill a cell. But actually, really importantly, NK cells also secrete a lot of what we call cytokines, which are these proteins that allow cells to talk to each other, to recruit other cells. So in this case, the NK cell is bringing in another cell to do its dirty work for him and remains fairly happy. And a side note, I did show this slide at my daughter's elementary school, and the principal was not terribly happy with me because she was worried that the kids would go out on the playground and imitate natural killer cells. So I'm now reserving it for adult audiences only. So this seems pretty simple, and we should totally be able to figure out the rules of this, right? And I should be able to make an NK cell vaccine that will kill the HIV-infected cells. Or so I thought 8 years ago. But there's always going to be a complication. And the complication is this is a massively oversimplified schematic of the potential interactions that might mediate this recognition. And so this is actually, honestly, from one of our vendors, R&D systems, where we buy antibodies to study these things. And this is the NK cell in the middle, and this is potential target cells. And these are known NK cell receptors. The pink ones are inhibitory, the blue ones are activating. And these are known ligands. And it turns out we don't know the ligands for a bunch of them, so we certainly can't figure out how they work. And so the question is, how does this work, and are certain ones important for certain situations? And to further complicate this, we decided to try and figure out, well, there must be certain receptor combinations that are important for things a few years ago. And so we started just looking, like if we look on the surface of an NK cell, what receptors are there? And it turns out like every possible combination. And so when we did this, we looked in healthy donors. We could define 120,000 NK cell subsets based on differential expression patterns of all these receptors, which is kind of neat, but kind of challenging. But it does really raise intriguing questions, because I told you earlier about the serial killers. We know that a minority of NK cells respond to a given target. So really the big question is which ones, and can we figure them out, and can we figure out how to control them? And of course, I wish I knew the answer, but I don't. So, so really, why do certain NK cells respond to a given stimulus? And this is an interesting area and really challenging, but it's also important to keep in mind that it's difficult in multiple ways and that there might not be a single answer. So these are, for instance, some of the viruses that and one bacteria that my lab works with. But it's important to keep in mind that the job of these pathogens, these are all pathogens, not commensals, is to escape. And so the ability of an NK cell to respond to the pathogen depends both on all those NK cell receptors I just showed you, but also on how the virus figures out how to mess with us to run away. And viruses and bacteria have shorter lifespans than we do. They evolve much faster. And so they have a competitive advantage in this evolutionary battle. And so just to go back to HIV for a minute, some of these pathways are known. So this is an example of an NK cell over here and an HIV-infected cell over here. And these are, through various people have done work, we've figured out some of the important signals that can allow recognition. And these are an example of 4 different HIV genes. HIV is such a simple virus. It's only got 10 genes. 10 genes, and it causes so much havoc, right? 4 of these genes encode escape mechanisms. It's kind of astounding. Or regulate NK cell recognition in some way. And the amazing thing about it is these are very small little proteins. They do it in different ways. So these are all things that are driven to be upregulated by HIV infection. But very importantly, these are all things that HIV genes downregulate to try and escape. And just to highlight one here, NEF is a simple little protein. It has 4 different ligands that might allow an NK cell to recognize it, that it pulls off the cell surface. And it does it not in the same way for each one of them. Some of them are early in the cell cycle, some are later. It's crazy. So this is what we're up against. And so it really makes you question, does NK cell activity even protect? And the answer is probably yes. This was a lovely study done by Daniel Scott Algira in people who were exposed to HIV but remained uninfected despite using drugs and sharing needles with the drug users. And so what we're looking at is, again, this was another old paper, so it's the best I could do. The HIV-infected unexposed. So these are protected people. So people who are naturally resistant to HIV, this is their ability to kill a tumor cell line, just the NK cells. Same assay I showed you from Christine Byron's paper. And what you can see is that they have really amazing NK cells. And we don't know if it's chicken or egg or what. But what's intriguing is if you look at healthy controls, they're better than just the normal person off the street. But even more importantly, these are people who didn't have HIV at the time of the study, but they went on to acquire it 12 to 18 months, within the next 12 to 18 months. So same IV drug using cohort. Some people went on to acquire HIV. Some people didn't in the 10 years that they followed these people. And they had this clear difference in activity. And you can see that once you get HIV, your NK cells kind of go to pot. So the question is really, how can we make everyone look like this, right? And if we think back to vaccines, we talked about this already. Most vaccines, all effective vaccines that are on the market, work through making antibodies. But what's been kind of interesting is in the last few years, NK cell memory has been described. So I told you earlier that the innate immune system doesn't have memory. So we now think that's wrong. And that at least, so this half shows experiments that have been done in the mouse. And there's clear memory to cytomegalovirus, a herpes virus. And actually, there's strong evidence for cytomegalovirus in humans as well. The hantavirus and chick and HIV and HPV are probably all non-specific CMV things because they are co-infected, so I don't buy it. But there's also memory-induced, cytokine-induced memory NK cells. So the question is, could we figure out how to program an NK cell to recognize HIV and do better if we got it later? And unfortunately, me and about 20 other people are working on this, and we don't have the answer yet. But it's also important to know that NK cells, again, do a lot. There's sort of a joke in immunology that all immunologists have their favorite cell type, and they put it in the middle of every graph and show how all the other cells circulate around it. So when my postdoc made this slide, I thought that was hysterically perfect. But you can see that the NK cells, they do the lysis, and if we could make memory cells, they could just knock off the HIV-infected cells. But they also have effects on these other cell types, T cells, macrophages, dendritic cells. And so we may not need memory. We may just need to sort of tune the response for enhanced responsiveness. So the question is, could we just swap it out and make NK cells? And unfortunately, big question mark, because we don't know yet. But we're working on it. But where we do know that NK cells have yielded very exciting results is in cancer. And so at the very conclusion, how am I doing on time here? I think I'm okay. NK cells play an important role in cancer, but NK cells can also be engineered to fight cancer. And so at the conclusion, I want to bring this up. So just like viruses, tumor cells try to escape NK cells, and they do this a whole bunch of different ways. So here we've got a tumor cell, here we've got an NK cell, and they'll try decoys, and they'll try to immunosuppress them, and all kinds of things. And all this occurs for T cells. How many of you have heard of checkpoint blockade? So a reasonable number. So these are the new drugs that have really revolutionized cancer treatment. And the idea is that tumors introduce what's referred to as a tumor microenvironment, where they basically induce immunosuppression, and they turn off all the immune cells that are there to kill the tumor. And all of these are mostly focused on T cells. And so the idea is you give these inhibitors that block the signals that the tumor gives to the immune cell to say bugger off. And so if you block the block, you unleash. Does that make sense? So it turns out NK cells express all these same things. And in some tumors, they have better access than T cells. And there are also other mechanisms. So just as a review, so here's PD-1. This is one of these major inhibitory signals. So these are the major drugs out. So if you just simply say, tumor cell, you can't talk to the NK cell or T cell and turn it off, the NK cell or T cell is more likely to kill it. Now, you can also, with NK cells, I already told you that class 1 molecules turn off NK cells. So there's a whole company, Innate Pharma, that is focused on basically blocking self in tumors to unleash NK cells and enhance their activity. And some of their targets are showing promise in early stage trials. You can also add antibodies. Rituximab is a classic example for lymphoma, to the tumor antigens. And NK cells have some of these receptors for antibodies, and they'll kill it. You can also unleash some of these activating receptors by inducing them, which is a little more complicated. And then finally, CARs. Everyone heard of CARs? Chimeric antigen receptor, CAR-T. So these are engineered receptors that have primarily been explored in T cells. They recently got FDA approval. And basically what you do is you hook a receptor to a tumor antigen with a specific reaction, and you use it to constitutively turn this on. So as soon as this CAR-induced NK or T cell sees its antigen, it will kill that cell. And this was originally done in T cells. The problem is they cause graft versus host disease. So it works great. Sometimes you want graft versus host disease because that can help you clear the tumor. Sometimes you don't. And NK cells, it turns out, don't cause as much graft versus host disease, so they might be better tolerated. So a lot of studies coming out. And then you can get really fancy with these and do bispecifics to bring in more things. So this is very early days, but I just got back from the meeting, but there's at least, I'd say, 30 ongoing clinical trials of these various things that we'll be seeing results of in the next several years. Okay, so I hope this has been an entertaining introduction to natural killer cells, and that you remember that they are early responders, they kill infected and tumor cells, and they're really critical for defense from viruses, particularly herpes viruses. But they're also promising candidates for immunotherapy, and I mean that broadly in terms of both vaccine and cancer therapy. And that's in part because they really blur these lines between innate and adaptive immunity, and that may provide us a window to figure out how to control them by tuning up or down these signals. So they are capable of memory, at least in some situations. They have tuned responsiveness. The array of receptors that are available on them can be turned up and down, and maybe we can use that to control their responses. And they're showing a lot of promise in cancer trials.